Classifying and identifying microbes is so important in medicine and in hospitals because it gives us an idea of what infection someone has and how to treat it. So in clinical settings, when someone has an infection, one of the most important things is determining the kind of organism that's causing the disease. And this involves two major steps. One is classification and the other is identification. And they're different things, but similar. So I will start with classification. And I'm using this in medicine, but you can use it in microbiology or with organisms in general. So starting off with classification, classifying an organism when you have an infection is basically looking at the big groups that an organism can fall under based on shared characteristics. So it helps us figure out what kind of microbe we're dealing with, whether it's bacteria or virus or a fungus. They're different things, or maybe something else entirely. These are big categories, and that's classification. So for example, when a patient has an infection, maybe you want to start off by saying, is it bacterial or is it viral? And this is important because treatments vary. Antibiotics can kill bacteria, but they're not effective against viruses. So a viral infection like the flu or a cold or COVID needs antivirals and supportive care, but bacterial infections might be treated with antibiotics. So beyond this broad classification of viruses versus bacteria, we can get more specific at determining the domain of life that the microbe falls under. Is it domain bacteria, domain archaea, or domain eukarya? And this classification is really important in microbiology because it influences how we understand the microbes' biology, its behavior, potential treatments. So for example, domain bacteria and domain archaea are both made up of prokaryotic cells, meaning they don't have a nucleus, but there's fundamental differences in their cell structure. So you may treat them differently, even though we don't really know anything about archaea infections, but that might change in the future. One important tool in classification in the big categories of microbes is something called rRNA sequencing. So ribosomal RNA is a component of ribosomes, and ribosomes are these things in our cells that make protein. And so in them, there's rRNA, and rRNA evolves so slowly. And because of that, it's a great tool for tracing evolution. And when you sequence rRNA, you can compare the sequences of rRNA. And with that, you can classify an organism as bacteria, as archaea, as eukarya. So that's why when we want to classify things in these domains, we could do something called sequencing rRNA. Okay, after we classify an organism, so let's say you classified your organism, you want to do something called identification. So classification groups organisms into these big categories, but identification is so important because it pinpoints exact species or the strain that's responsible for the infection. So identification is what's important in hospitals and clinical settings because it allows you to decide on the treatment for the specific pathogen. To give an example of identification, so for example, when a patient has pneumonia, it's a bad lung infection, the first step is to classify the infection as bacteria or viruses because bacteria and viruses can cause pneumonia and other things as well. We need to identify the exact bacteria so we can look at whether it's like streptococcus pneumonia or something else. And this identification is so important because different bacteria can require different antibiotics. It's the same example with UTIs, urinary tract infections. We can classify the infection as bacterial, but then we need to decide what type of bacteria is causing the UTI. Is it E. coli? Is it Klebsiella? Is it something else? So you can decide on the best antibiotic to give. And there's many different ways that we can identify microbes. So microbes, we can identify them in two ways. We can do phenotypic identification. That's looking at physical characteristics, and then genotypic identification. So I'm going to talk about phenotypic identification. Phenotypic identification is you got the patient's urine sample, and now you're going to look at it under the microscope. You're going to do a gram stain. You're going to look at it on an agar plate, and you're going to do various metabolic tests that you see with your eye. and you get all this knowledge together and you say, because my bacteria from the urine is gram positive and it's a rod and it's motile and it's positive for fermenting sugars and it has catalase and it doesn't have oxidase, you know exactly what it is. That's phenotypic identification. Now, in these years, a lot of times we do genotypic identification. This is looking at things like DNA sequencing or PCR. PCR has to do with looking at genes or DNA, and we can identify the exact bacteria it is. This is so important for fast identification where you need to know exactly what to do. So for example, if you have antibiotic resistant bacteria, knowing exactly which strain is involved helps so that you don't prescribe wrong antibiotics. So overall, both classification and identification are so important in diagnosing infections and making sure that people receive the best treatment. And by first classifying the organism, whether it's bacteria or viruses or a eukaryote or something else, we get a general type of understanding. And then by identifying the exact species or the strain of the virus, you can fine-tune the treatment to target the pathogen more effectively. Now, all of this classification and identification, this is going in a different direction now. It's super cool, but looking towards the future and now, artificial intelligence can now help with microbial classification and identification. So these technologies have so much potential to look at very large data sets like genetic sequencing or metabolic tests. We call those metabolic profiles, much faster than just having a person look at them. And when you get all of that, AI can spot different patterns and relationships to better quickly identify an organism. So for example, you can have an AI that is developed to analyze medical images like microscopic images. You can add genetic data. You can put all the results of your metabolic tests, and the AI can classify and identify your microbe based on all of these things. So that's classification and identification. And in medicine, we care about identification. Both are important. Classification is big categories, and then identification is knowing the exact strain or species. So one last time to say this. So classification meaning that someone comes to you and you're trying to determine is what they have, a virus, bacteria, eukaryote. And then identification is what type of virus do they have? Which bacteria do they have? So that's classification and identification.